the entire human genome has been sequenced and it is stored in their database of that particular program. So satellite DNA is nothing but, depending on base composition, they can just check whether it matches with the computer system file they have. So that is how they find out the criminals. The technique of DNA fingerprinting was first developed by Alec Jeffreys. Hello everyone, a warm welcome to this session for Class 12 Biology. I am Dr. Divya, Biology faculty. So in this session, that is the last session of Chapter 5, Molecular Basis of Inheritance, we will learn about DNA fingerprinting and the technique that is followed in DNA fingerprinting. So in the previous session of this chapter, we had learnt about Human Genome Project, right? So in Human Genome Project, we got to know that the entire human genome has been sequenced and it is stored in their database of that particular program. And we also got to know about the repeated sequences. So when we say sequencing of the entire human genome, means the number of repeated sequences also we know about it. And hence, in this particular topic, we will continue with learning DNA fingerprinting. So I had told you in the previous session that the number of repeated sequences in each person tends to vary, and that is actually a boon to the human community, and that is why we are different from each other. So though we have a set of genes which are quite similar and their function is to produce the same kind of proteins, but still because of the variations in the number of repetitive sequences that are there on a DNA strand, each of us tend to vary genetically and they are unique from individual to individual, and that actually helps in DNA fingerprinting technique. So DNA fingerprinting technique involves identifying differences in some specific regions in the DNA sequences, which is called as repetitive DNA, because these repetitive DNA sequences are nothing but a small stretch of DNA that gets repeated many number of times, and the repetitions can vary from one person to another, or there can be some alteration in the gene during the repetition process also. And these repetitive DNA are separated from the bulk genomic DNA as separate peaks during density gradient centrifugation. So if you remember, we had learnt that in while DNA sequencing, what they do is, so in the DNA sequence, they have the coding regions as well as the non-coding regions also, right? Non-coding genes as well as the coding genes. So some of these repetitive sequences that are there, they do not code for any proteins, and that is why if they vary from individual to individual also, it is not going to alter any of the normal functioning of the human body, because they do not code or produce any kind of proteins. But the coding genes which are there, they code for a number of proteins and they should not get mutated. And if mutation occurs in the repetitive sequences also, it is fine because it can lead to evolution. Other than that, it will not cause any problems in the human beings or any of the organism for that matter. So these repetitive DNAs which are there in between the coding regions of the DNA strand, they actually can be separated. So how do we get to know about it? So we had studied, right? So wherein the chromosomes will be isolated from the cells of the individual, and that chromosome will be subjected to various centrifugation using various enzymes. And after the centrifugation process, we get the data that will be read in the computer. Now the data can give you the nucleotide sequence also, and it can also give us the number of repeated sequences that are present in that particular DNA strand. How can we get to know that? It is because of different density gradation. So when this particular DNA is subjected to different density gradation technique, we will get lot of high peaks like this. So wherever we have very high peaks, we can say that, okay, the number of repetitive sequences in that particular DNA strand is 1, 2, 3, 4. So, how many in that particular? 4. So, some individuals might have 4, some individuals might have more. So, it varies from individual to individual. It is in this point, the repetitive DNA is separated from bulk genomic DNA as different peaks during density gradient centrifugation. The bulk DNA forms a major peak, and other small peaks that are there, they are called as the satellite DNA. Now, these we can call as the satellite DNA. The other ones which forms the highest peaks, they are called as the repetitive DNA. And always remember, the repetitive DNA or the number of repetitions or number of repetitive sequences in the DNA is unique to one person to another, and it varies from individual to individual. So because we learnt about, now you know what is repetitive DNA, we learn about satellite DNA, because in that sequence of DNA, there are repetitive sequences as well as there are satellite sequences also, satellite DNA. So what is this satellite DNA? So satellite DNA is nothing but depending on base composition. So when I say base composition, I am talking about the nitrogenous bases. And in DNA, we know that the nitrogen bases are adenine, guanine, cytosine and thymine. Depending on how much adenine, how much guanine, how much thymine and how much cytosine is present, the length of the segment and number of repetitive units, the satellite DNA is classified into many categories such as microsatellites and mini satellites. So again, how many times the nitrogenous bases has repeated on the DNA strand, based on that the satellite DNA can be classified into microsatellites and mini satellites. So these sequences, they normally do not code for any proteins, but they form a large portion of the human genome. So the satellite DNAs also do not code for any proteins, just like the repetitive DNA, and these sequences show high degree of polymorphism. The repetitive sequences of satellite DNAs, they show high degree of polymorphism. Polymorphism means they are different from individual to individual. Every individual, it is unique, and they show different characteristics when compared to one individual. They are the same genes which control different characteristics in an individual. And therefore, they can be widely used in DNA fingerprinting because they are unique to individual. So, you learn about that in detail when I talk about the technique that is followed in DNA fingerprinting. So, now talking about DNA fingerprinting. So, since DNA from every tissue, so DNA can be isolated from almost every tissue, even a small bit of hair strand, a small drop of blood, the hair follicle. Even if we get not just the hair strand, even the hair follicle if we get also, we can isolate DNA out of it, from the skin, from the bone, from the saliva, sperm, etc. Any of the cells that are there, DNA can be extracted, and they show some degree of polymorphism, of course, right? Because say for example, there is a person A and there is a person B. Person A has different set of satellite DNA in them or repetitive sequences in them, and person B has different set of repetitive sequences. The coding genes are same in both. Okay, I'm talking about the non-coding genes. That is, non-coding genes which are present in the repetitive DNA of both the individuals. Okay, so here they exhibit polymorphism. Say for example, the number of repetitions of a particular nucleotide in one person or repetitive sequences in one person is 11, and the person B, it is 10. Are they not different? They are different. So here, but the number of repetitions, be it in person A, the number of repetitions of the repetitive sequences in the DNA extracted from the blood, hair follicle, skin, bone, saliva, sperm will be same. Same in person B also, it will be the same. It will be the same means if he has 10 repetitive DNA strand or repetitive DNA sequences, if he has 10 repetitive DNA sequences in his hair follicle, skin, bone, saliva, sperm and all that, the person B will have 11. So it's like that. So all the cells will have 11 repetitive DNA sequences itself. So that is about this particular uniqueness of the repetitive DNA sequences. So since DNA from every tissue of an individual shows same degree of polymorphism, they become very useful as an identification tool in forensic application. So how we will study later. So further, as the polymorphisms are inheritable from the parent. So repetitions are inherited from the parent. Like sometimes the number of repetitive sequences that are present in the parent can be inherited, can move on to the daughter or move on to the progenies also. So as the polymorphisms are inheritable from the parents to the children, DNA fingerprinting can also be used to identify the biological father, the biological mother of the child. So, DNA fingerprinting can not just help catch criminals but not just help in forensics, but it also helps in genetic related issues wherein does that child belong to the mother or to the same father? Who is the biological mother of the father can be found out using this DNA fingerprinting technique. And polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as DNA fingerprinting. Because I told you in human genome, each of our genome tends to vary because the number of repetitive sequences that are there varies from individual to individual. So, if all the data sets, say for example, there is a criminal who has already a background of doing a crime. So, already his DNA database will be there in the, or his entire genome sequence will be there in the system. Next time, if they have a doubt whether it is the same criminal who has conducted a crime, there is no need for again isolating the DNA and doing studies on him. They can just check, or they can just check whether it matches with the computer system file they have. So, that is how they find out the criminals. So, this is about DNA fingerprinting. So, talking about, so everywhere there was the word that kept on repeating called as DNA polymorphism. Now, what is this DNA polymorphism? Polymorphism. Poly means many, morphism, morphological. We have many morphological characters which are not exactly same to our parents. Some which are same, some which are not, right? So, that is about DNA polymorphism. So, DNA polymorphism is an inheritable mutation and is observed in a population at high frequency and is referred to as DNA polymorphism. So, these mutations will not cause any problem to the individual because they do not code for any of the proteins. They are not the coding genes and therefore they do not cause any problem. But these mutations are needed in a slower rate for the evolution process to take place. They are needed. And these mutations keep on accumulating generations after generations and form one of the bases of variability or polymorphism. That's true, right? That is how we might have evolved from the monkeys, because these mutations that happens on the repetitive sequences that resulted in polymorphism. Now, are we just exactly like the monkeys? No, mutation results in evolution. So, we are evolving. We have evolved from the monkeys and still we are still evolving. It is because of this polymorphism conditions or the mutation that occurs on the repetitive sequences. And there is a variety of different types of polymorphism ranging from single nucleotide change to very large scale changes. All this you have studied in point mutation, frame shift mutation, right? So, if not, you can have a look at that topic or that session wherein we learnt about the mutations while dealing about genetic codes, like what happens if mutations happen. There are two types, point mutation and frame shift mutation. So, how alterations can happen, all that you have learnt in the previous session. So, and for evolution and speciation, such polymorphism play a very, very important role. So, therefore, polymorphism not just helps the human race to have an identity of their own, but also for the evolution to take place or for speciation to happen, it is very, very important. And also it is important because it also helps the organism to survive in a particular habitat, because they can alter themselves to make sure that the organism can suit to that particular habitat. So, for all that, these DNA polymorphism is very, very important. And this DNA polymorphism is one of the major factors that helps in DNA fingerprinting technique. The technique of DNA fingerprinting was first developed by Alec Jeffreys, and he used a satellite DNA. What is that satellite DNA? So, satellite DNA are nothing but again repetitive sequences which consist of tandem repeats. That is, they contain non-coding genes on their strand. So, they are the satellite DNA. They used the satellite DNA as a probe that shows very high degree of polymorphism. So, those probes which shows very high degree of polymorphism, because sometimes polymorphism will not be much. It will be exactly similar to our parents. So, we should not choose such strands for the study. So, strands which show a high degree of variability should be used for the study. And it was called as variable number of tandem repeats. So, the technique as used earlier, it involved southern blot hybridization using a radio labeled VNTR, that is variable number of tandem repeats, as a probe. So, what they do here is, we'll just have a look at this. So how it is done? So this is, imagine a crime has happened somewhere. Somebody has killed someone, and when they killed that person, during the fight, both the culprit and the culprit also had to shed some blood. So a drop of blood just fell, or a drop of blood was also there on the knife that he used to stab the victim. So that the forensic people, they'll go there, they'll remove the blood sample, they'll take it to the lab and they will start investigating. So now in that blood sample, there are RBCs which are present. That's also a cell. So these RBCs, from the blood sample, they'll isolate the DNA. Now when they isolate the DNA, or from the hair follicle or from the nail, whatnot, they can isolate the DNA. So now the DNA, so they have mentioned here DNA from individual A and DNA from individual B. So they have suspicion on 2 individuals. So they are doing or mapping the chromosomes of 2 individuals, that is DNA from individual A and DNA from individual B. So here, after mapping, what they got was, they saw the chromosome. They did the sequencing of the DNA from the blood sample that they got, and already this is done and it is stored in the system. Okay, for example, this is done and stored in the system. So here, the person A is having the paternal chromosome and the maternal chromosome. That's true, right? If an individual, say for example, I will have 1 chromosome from my mother, 1 chromosome from my father. That is true, because you have learnt in sex determination, how sex determination is done, right? Wherein we get 1 chromosome from the father, 1 chromosome from the mother. We don't get both the XX chromosome from the mother, or we don't get both the X and Y chromosome from the father. If we get 1 X chromosome from the mother and 1 X chromosome from the father, then the fetus will be a girl. If X chromosome from the mother and Y chromosome from the father, then the fetus will be a male, right? So we get both the chromosomes from the paternal, that is from the father, and maternal from the mother. So if you see here, paternal chromosome had 1, 2, 3 repeats, repetitive sequences, and maternal chromosome has 4 repetitive sequences in individual A. In individual B, the paternal chromosome has 11 and the maternal chromosome has 9. If you count, you will know it, 11 and 9. Isolated the chromosome and then they did the DNA sequencing, and then they got to know that the number of repeats in paternal chromosome of individual A is 3 and maternal chromosome is 4. The number of repetitive sequences on individual B's paternal chromosome is 11 and maternal chromosome is 7. They got to know. Now in chromosome, this was in chromosome number 7. Chromosome number 7. Then they isolated, then they checked the chromosome 2, because in a cell inside the nucleus will have 46 chromosome. So they checked the second chromosome. In the second chromosome, they found out that it is having 1, 2, 3, 4, 5, 6, 7, 8. Here it is 8 and here it is 7. Again here 1, 2, 3, 4, 5, 6, 7, 8. 8 and it is 7. In chromosome 2 of individual A and individual B. Again chromosome 16 they took, because they are seeing where too many repeats are there, those chromosomes they are choosing. Okay. So here they found out that in chromosome 16, 1, 2, 3, 4, 5. 5 repeats are there in paternal chromosome, that is chromosome which came from the father, and 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 repetitions. Here in individual B, it is 1, 2, 3, 4, 5, 1, 2. So they have now 2 set of DNA sequences of different chromosomes, that is of chromosome 7, chromosome 2, chromosome 16 of individual A separately and individual B separately. Now they know how many repeats are there or how many satellite DNAs are there in this particular chromosomal strand. They know now, right? After that, what they will do is, now they will take this and they will put it on a gel electrophoresis. That gel electrophoresis, they'll cut the DNA strands, they'll cut the DNA strands into bits and they will put it in gel electrophoresis. When they run in gel electrophoresis, they'll give the electric currents. So that electric currents, when they push, the DNA strands which are heavy will move the slowest and the DNA strands which are light will move the farthest. And based on that, the strands will get repeated. Now, if you can see here, and they use radioactive probes. probes will give coloration. Otherwise, we can't see chromosomes in different colors. It is difficult. Or DNA strand in different colors. So, we can see different colorations here. Now, when they count here, now this is one chromosome. This is also one. So, they are amplifying. Amplifying means they are increasing or they are cloning the DNA, because with small DNA strands, we cannot do any study. So, they make clones of the DNA strand and then they use it for this experiment. Once they are separated, they will remove it. If you can see here, they are removing it. Right? All this. Now, they wanted, this is chromosome 7 of individual B. This is chromosome 7 of individual A. Now, if you can see here in this chromosome, like that they have separated. Okay, so this was the DNA that they got from the blood sample in the crime scene. These are the suspects. They'll have some doubt, no, their chromosome sequencing or DNA sequencing has been done. This is from the crime scene, whatever blood they got. Now, if you see here, you can see again it has how many repeats? 11 repeats. Here there are how many repeats? 9. Here again it is 8, it is 7. Here again it is 5 and it is 2. Now tell me, with whom is it matching? It is matching with the DNA from individual B. So therefore, who is the suspect? It is the individual B who is the actual culprit who actually created that crime. So therefore, they will get to know about this. So this is about the gel electrophoresis technique that is actually followed for DNA fingerprinting. So this is the gel electrophoresis technique that is followed for DNA fingerprinting. So this is how, by using, sometimes in the police station their records itself they'll have, or in the forensic department records itself they'll have all these chromosomal genomic sequence of the individual A and individual B. If both were criminals before itself, they would have. If it is a fresh criminal, they'll have to isolate the DNA and see the sequences and all that. Otherwise, it will already be available. So let's talk about the details. So first is isolation of DNA, which I have told you. Then the DNA needs to be cut at certain regions, and to see, that is in the repetitive regions, it needs to be cut. So that is done by using restriction endonucleases enzyme. Then the DNA fragments that are there should be separated based on the density. That is done on gel electrophoresis. Then the DNA strands that are there, it will be transferred to a, which we call it as the southern blotting technique. They will be transferred to a synthetic membrane. That synthetic membrane will be made up of nitrocellulose, or it will be made of nylon, because you can't use that from the gel. The synthetic material, it will be kept on the gel electrophoresis so that the DNA gets like a blotting sheet, so that the DNA strands can go and stick onto it. After that, hybridization will be done using a VNTR probe, because we can't see the DNA. So therefore, different probes will be sent. So whichever probe is matching or complementary to that particular sequence, no, that will go and bind there, and it will make, and when we put that slab, that is a nylon sheet under the UV radiation, different colors we can see. So that will be done, and then detection of the hybridized DNA fragments by autoradiography, as I said, that will be done, wherein we can find out. Now we can see, then it will again, these sequences will be subjected to bioinformatics studies, wherein we can get to know the number of repeats that are there. Based on the number of repeats, we got to know that the culprit was individual B and not individual A. So that is how DNA fingerprinting is done. So let's learn about what is this VNTR probe. VNTR, variable number of tandem repeats. So variable number of tandem repeats belong to a class of satellite DNA, which is referred to as mini satellite, based on the number of AT and GC rich nucleotides that are present on the DNA strands or the repetitive sequences. The repetitive sequences or the satellite DNA are of different types. Mini satellites and microsatellites were learned, right? So the variable number of tandem repeats, they are an example of a mini satellite. A small DNA sequence is arranged tandemly in many copy numbers. All this I have told. The copy number varies from chromosome to chromosome in an individual. That's true, right? Copy numbers varied from chromosome to chromosome. If you see here, in chromosome 7, the repetitive or VNTRs are different. In chromosome 2, it is different. Chromosome 16, it is different. So that is about VNTR. The number of repeats show very high degree of polymorphism, and as a result, the size of VNTR varies from 0.1 to 20 kb. So that is about, and based on the size, they get separated on the gel electrophoresis plate. And when electric currents are passed, the ones which are lighter will travel the farther, and the ones which are heavier will travel the slow, will travel very short distance, and for that VNTR probe. So VNTR probe means, say for example, the tandem repeats in that is ATGC, ATG, AG, like this. Now for that probe will be made, which will be radio labeled. Radio labeled means when put, it can illuminate light. So for this complementary will be there. It will be T, A, C, T, A, C, T, C, like this. So when we send complementary, they will go and bind there, because complementary strands and all that you have written. So such probes will be designed, n number of probes, in that whichever is complementary will go and bind there, and therefore it will fluoresce, or it will emit some radiations or emit some colors. So that will be again sequenced, and then they will find out actually what is the number of repetitions that are there. So this is about VNTR. So now let's learn about what are the applications of DNA fingerprinting. So DNA fingerprinting is used in forensic science to understand or to find out who the exact criminal is, and it is used in determining population diversity. So because the number of tandem repeats or variable number of tandem repeats or satellite DNA or repetitive sequences, whatnot we can call them, they vary from individual to individual. So, and also it varies from habitat to habitat of that individual as well, and to determine genetic diversities, because they are the main source for evolution, and evolution is important for speciation also, because evolution will give rise to new species. So to understand the genetic diversity also, this DNA fingerprinting is very, very important. So this was about the DNA fingerprinting. So I hope this session was quite useful and it was quite interesting. So with this, the chapter comes to an end. So we shall meet again in the coming sessions taking up a new chapter. So see you in the coming sessions. Thank you.